Hello everyone, welcome back to the session. So in today's class we are going to study about the short answer question in the chapter called the locomotion and reproduction in protozoa. Okay, so the first question is, name the system that controls the fast swimming movement of protozoans and write its components. Okay, so first we have to name that what is the system that is controlling fast swimming movement. Okay, so and that system is called as an infraciliary system. Okay, so that is the name of the system and how it is involving in controlling the fastest swimming movement in case of protozoans. Okay, so the name of the system will be this infraciliary system, where it is exactly located. It is located just below the pellicle. So you should know what is meant by pellicle. So pellicle is an covering, outer covering layer which is present only in the protozoans. Okay, so that is a pellicle. So below the covering, that is called as a pellicle, we can see the system which is called as an infraciliary system. Okay, so we are having ectoplasm and endoplasm, but here it is in the ectoplasm of the ciliates. So that is what the location of the infraciliary system. So what are all the components present in this system? Kinetosomes. Okay, so the kinetosomes is nothing but this is the structure which gives rise to cilia. Okay, so it is located at the base of the cilia in a transverse and longitudinal rows. Okay, so longitudinal in the sense this is what a longitudinal, and transverse in the sense this is the transverse rows. Okay, so this kinetosomes is located at the base of the cilia, both in transverse as well as the longitudinal rows. And what about this kinetodesmal fibrils? Okay, so this is what are connected to the kinetosomes. Okay, so this is connected to the kinetosomes. It runs along the right side of each row of the kinetosomes. Okay, so this is very important. This kinetodesmosomal fibrils, this is connected to the kinetosomes. Okay, so this is run along the right side of each row of the kinetosome. So this is the kinetosomes, this is the right side. So in the both transverse rows as well as the longitudinal rows, in both rows we are having the kinetodesmal fibrils. Okay, now a cord of fibers, a cord of the fibers which is a cord of fibers formed by the kinetodesmal fibril. So that is what a kinetodesmata. See, kinetodesmata, this is the fibers, is a cord of fibers that is connected, that these are all the fibers formed by the kinetodesmal fibrils. Okay, so then what is kinety? Okay, the longitudinal rows of kinetosomes together with kinetodesmal forms a kinety. Okay, so a longitudinal row of kinetosomes together with the kinetodesmata forms a kinety, which means what? See, this is the kinetosomes. The kinetosomes, they are having they together with the kinetodesmata. So they with kinetosomes together and the kinetodesmata together, we are calling them as a kinety. Okay, motorium. It is located near the cytopharynx. Okay, the pharynx structure is here. So this is the structure which is present near the cytopharynx. Okay, so this motorium is also connected to the infraciliary system. Okay, so what is the function of this infraciliary system in case of the swimming? Okay, so all kinetics together form the infraciliary system. So you should know that all the kinetics, which mean kinety, we have studied that all the kinetics together form the infraciliary system. Now infraciliary system plus the motorium. So this motorium is a structure present near the cytopharynx. So together we are calling them as the neuromotor system. Okay, so this system controls and coordinates the ciliary system, enabling the fast swimming in the protozoan. Okay, so you should know that neuromuscular system, as the neuromotor system is involving, the ciliary function is in the coordinative manner. Okay, so this system is the one controlling as well as coordinating the movement of cilia. So this is the one enabling the fast swimming in the protozoans, right? So that's all about the first question. The second question will be, write the mechanism of bending of flagellum and explain the effective and recovery strokes. Okay, so what are all the protein which is involving in the flagellar movement? Okay, dynanorms as well as the ATP. Okay, so dynein arms exhibit the cyclical movement using energy from the ATP. Dynein arms are the sites of the ATPase activity in cilia as well as the flagella. Okay, so you should know that dynein arms. So dynein arms is nothing but this arms is made up of a dynein protein. That is why the name dynein arms. So this is the one exhibiting a movement and which is cyclic in nature. So that is what a cyclic movement using an energy. For this cyclic movement done by this dynein arms requires energy in the form of ATP. Okay, so this dynein arms are the sites of this ATPase activity that is in the cilia and the flagella. Okay. So the dynein arms are the sites of this ATPase activity where exactly the requirement of energy is taking place, that is both in cilia as well as the flagella. Now the microtubule interaction. Dynein arms and one microtubule attach to the adjacent doublet and pull it, causing sliding in opposite directions. Okay, so dynein arms which is present in one microtubule attaches to the adjacent doublet, which means that microtubule which is always exhibit in doublet. Okay, so that is what one microtubule, it attaches with the another microtubule doublet and it is going to pull it. Okay, one microtubule is going to attach with the another microtubule and it is pulling the other microtubule. This pulling action causes the sliding in the opposite directions. Okay, so when these dynein arms releases and reattach further along the adjacent doublet and repeat the pulling action. So when you should know that this dynein arms is going to attach with the microtubule, only then it is going to pull the microtubule. So this pulling action causes a sliding movement in the opposite direction. So you should know that this microtubule interaction is very important for the movement of the cilia, and for this microtubule interaction there will be an involvement of this dynein arms. Okay, so this requires ATP. Now what is the point of having this radial spokes and bending? Okay, so this radial spokes prevent the excessive sliding by anchoring the doublets, resulting in bending instead of full displacement. Okay, so the radial spokes is a structure that is the one preventing the excessive sliding. So which means excessive sliding by anchoring the doublets, resulting in bending instead of full displacement. Sometimes the animal doesn't need, the microorganism doesn't need the movement, but instead of that it just needs a bending. Okay, so for that it will be interacting in the microtubule interaction. It is interfering in the microtubule interaction. So that is why this radial spokes is preventing the excessive sliding done by the microtubule interaction. How it is preventing the excessive sliding? Just by anchoring the doublets. Okay, so just by anchoring the doublets it is preventing the excessive sliding, and it results in bending instead of full displacement. Full displacement of microorganisms has been prevented just by the radial spokes. Okay, by preventing the excessive sliding. Then this prevention results in the bending of an organism. Okay, so this bending of an axoneme causes the flagellar or ciliary movement essential for locomotion. So this bending of an axoneme, so this is very important, and this is very essential in the locomotion function. Okay, so this is what a microtubule doublet. So here one, here one. Okay, so these are all the microtubule tube doublets, and this is the arm which is made up of a protein called dynein arm, so dynein protein, so that is why the name dynein arm. Okay, so it needs an ATP for doing the ATPase activity, and there is an so this is where the ATP is going to bind, and ATP hydrolysis is taking place by the ATPase enzyme, and the anchorage in the cell, this results in the bending of the microtubule. So this is very important. Right, now the question has also been asking, explaining the effective and recovery strokes. Okay, so the flagellar stroke types, the first stroke will be the effective stroke and the second stroke will be the recovery stroke. What is meant by effective strokes? Here the flagellum becomes rigid and bends to one side. Okay, so you should know in the flagellum here it is it is not flexible here, it is rigid, and it bends to one side. It beats against the water at a right angle to the body axis, propelling our organism forward. So you should know that it is beating the water against it is beating the water against the flowing of water. Okay, so it makes, this is called, this is why the energy is needed. So in the stroke, so that is why we are calling the stroke as an effective stroke. In this effective stroke, you should know, as it is beating against the water, the organism is moving forward. Okay, and the recovery stroke in the sense, the flagellum here, it is not rigid, it is very soft. Okay, it is very soft, offering the minimal resistance to water. Okay, there is minimum resistance to water. It is not beating the water against the flow, okay, against its flow, but instead it is just moving on where the through the water is flowing. Okay, so that is what it is, minimum resistance to water. It returns to its original position, completing the stroke cycle. Okay, so recovery stroke in the sense, it is returns to its original position. So effective stroke in the sense, it is moving the water, the organisms moves forward. Okay, so it is moving back to its original position in case of recovery stroke. Okay, so it's thus completing the stroke cycle. Okay, that's what the second question. And the third question is, describe the process of transverse binary fission in case of paramecium. Okay, so paramecium, the process of here the question is demanding the process of transverse binary fission in the paramecium. Okay, how it is initiating, occurs under the favorable conditions after the paramecium attains the maximum growth. Okay, it is feeding activity stops before division begins. So initiation in the sense occurs it under favorable condition, only in the favorable condition. So here the transverse binary fission is nothing but a reproduction. Okay, so it is a reproduction function. It is occurring under the favorable condition. Okay, so in the favorable conditions, they are undergoing a binary fission, that too transverse binary fission occurs under the favorable condition after the paramecium attains the maximum growth, once it is matured. Okay, so that is called the maximum growth, that is sexually matured. Okay, so that is what the paramecium undergoes a maximum growth. Feeding activity stops before the division begins. Okay, if the feeding activity stops before the division begins, or before the division, before, once the binary fission, once the paramecium is ready to undergo this transverse binary fission, it stops feeding. Okay, the feeding activity is abruptly stopped there, and after that it is nuclear division. The nuclear division will take place, and here there is two different types of nucleus in case of paramecium. Okay, so that is what dinucleic, micronucleus and macronucleus. So the micronucleus is divides by the mitosis, and the macronucleus is divided by the amitosis. Okay, so micronucleus, this is very important. We are having two different types of cell division, mitotic division and meiotic division, but here it is amitosis and mitosis. This micronucleus is related to the it is going to do the reproduction function. This macronucleus is involving in the vegetative function. Okay, so the reproduction division will be completely taken controlled by this micronucleus. So that division is called as a mitotic division. So this macronucleus is undergoing amitotic division, just it is going to divide into two nuclei, that is two daughters, similar nuclei, just like that a parent nuclei. Okay, now oral group disappearance, whenever the transverse binary fission is taking place, all these modifications are undergoing in the body of the paramecium. Okay, so the oral group disappearance, the oral group disappears temporarily during the division. Okay, so the oral group is a structure. So this is what 1 minute. So this is what a paramecium. So this structure is called as an oral groove. Okay, so this oral groove is disappearing temporarily only when the division is taking place. Okay, and the second thing will be the cytoplasmic division. Okay, so always the nuclear division starts first, and after that followed by the cytoplasmic division. So this nuclear division is called as the karyokinesis, and the cytoplasmic division is called as the cytokinesis. It is not karyokinesis, it is cytokinesis. Okay, so the transverse constriction forms in the middle of the body. So this constriction deepens and splits the parent cell into two daughter individuals. So that individual we are calling them as the proter and ophist. Okay, proter is nothing but an anterior daughter and ophis is called as the posterior daughter. See, this is what are stages of the transverse binary fission. See here, this is the macronucleus. The bigger nucleus is called as a macronucleus and the smaller nucleus is called as a micronucleus. Okay. So now this is the one undergoing. First thing will be the disappearance of the parent oral groove. Okay. So this parent oral groove is getting disappearing here. Okay. Now this micronucleus is undergoing a mitotic division and the macronucleus is undergoing the micronucleus is undergoing a mitotic division. Macronucleus is undergoing an amitotic division. Okay, so this is what, after the disappearance of this oral groove, the new oral groove is getting generated. Okay, so that also the new contractile vacuole is also producing here. So as a result, so this anterior portion is getting developing and the posterior division is also developing. Okay, so this anterior portion is called as a proter and the posterior portion is called as the ophis. So this is what are stages of this transverse binary fission in the paramecium. Right now, so what are all the organelle distribution, what are all the proter is going to receive, what are all the ophis is going to receive. Okay, so the proter is receiving the this is the anterior contractile vacuole, cytopharynx, cytostome from the param, because it is an anterior position. No? So in the anterior position all the mouth region is going to going to be present. Okay, so develops new posterior contractile vacuole with an oral groove. Okay, so the ophis receives posterior contractile vacuole from the parent. Again, it is also going to receive from the parent. See here, from the parent, this proter is receiving the contractile vacuole, cytopharynx and the cytostome. This ophis is going to receiving the contractile vacuole from the parent. It is going to develop the new anterior contractile vacuole, cytopharynx, cytostome and the oral groove. These are all the structures. Okay, the proter is going to receive all these structures and ophis is also going to receive all these structures, right? So the completion and the rate. Okay, binary fission completes in about 1 minute. Binary fission completes in about 2 hours. Okay, so this completion, in this completion rate, the binary fission completes in about 2 hours. It takes 2 hours to reproduce completely, and this paramecium is producing 4 generations per day under the favorable condition. So this is an ideal condition. So in an ideal condition in the sense, there is what a favorable condition. So in this favorable condition, the paramecium can reproduce 4 generations per day. Okay, so each generation is going to take 2 hours. So what are all the important terminologies? So this transverse binary fission is also called as a homothetogenic fission. Okay, and fission, if the fission plane is at right angles to the longitudinal axis, we are calling them as the homothetogenic fission. Okay, so and it is also called as a perikinetized fission. So this fission occurs at right angles to the kinetics. So this is very important. If it is right angle to the kinetics, it is called as a perikinetized. If it is right angles to the longitudinal division, it is called as homotethogenic. Okay, so this is an important thing you have to remember. Now the next question will be this: describe the process of longitudinal binary fission in euglena. Okay, so in the previous question we have discussed the process of transverse binary fission in case of paramecium, and here it is the process of longitudinal binary fission in euglena. Okay, so now this is the initiation of the division. Begins with the division of nucleus, basal granules, chromatophores and cytoplasm. Okay, how the division is beginning. So there is a beginning with the division of nucleus. So nucleus is going to dividing, and the basal granules is dividing, and the chromatophores and the cytoplasm. So the initiation of division, the division is initiating by the involvement of all these structures. First, there is a nuclear division is taking place. The nucleus is divided by this mitosis. Nucleus is undergoing a mitotic division. As a result, it is going to give rise to 2 daughter nuclei. So the organelle division. So in the organelle division, the kinetosomes and the chromatophores also undergoes the division. So this kinetosomes, this is the one for the flagella. Flagella is going to arise from the structure called the kinetosomes. And the chromatophores, this is the one responsible for doing photosynthesis in euglena during the daytime. Okay. Okay, so undergoing a division. So this is what an organelle division, and this is the longitudinal binary stages, binary fission stages in case of Euglena. So first, initially the nucleus is getting divided. Okay, nucleus is getting divided, and this is a chromatin, and the nucleus is getting, the chromosome is getting duplicated. Okay, so the new chromosome is getting duplicated, so that is what a nuclear division. Okay, now the nucleus is elongating. Okay, so before it is completely dividing, so the nucleus is elongating. Okay, so this is finally there will be a daughter euglene. Okay, so these are all the important stages. Okay, the nucleus, so initially it will be like this, and after that it will be like in an elongation stage, and finally it will be divided into two different nuclei. So this is an important stages. Okay, this now, so this structure is called as a groove. Okay, so this is a groove. The longitudinal groove appears at an anterior end. The groove extends posteriorly, gradually separating the two daughter nuclei. See, initially it will be only one euglena, and finally it will be like this. Okay, so this is the structure called as a groove, right? So initially it will be at the anterior end and followed by this posterior end, gradually separating two daughter cells. Okay, flagellar distribution. Here one daughter euglena retains the parental flagella. Okay, so one daughter euglena, this is the one responsible for retaining the parental flagellae, because in the one single body it gives rise to two. Okay, so as a result, one daughter nuclei will develop their the flagella of their own, but one euglena will retain their flagella from their parent. Okay, so this is what a flagella distribution. So the other develops the new flagella from newly formed basal granules. So this is very, very important. From the newly formed basal granules, the kinetosomes will be produced, and from the kinetosomes a new flagella is going to produce. Now, organelle regeneration. Okay, organelle regeneration, stigma, paraflagellar body and contractile vacuole of the parent will be disappearing. Okay, so stigma, paraflagellar, contractile vacuole of the parent, these are all disappearing. Okay, so they disappears. These structure develop afresh in both daughter cells. Okay, so these daughter cells are going to develop newly. Okay, these are all very, very important. These are all the important structure that developed newly. It is not depending on the parent. Okay, so what are all the types of division? So it known as the symmetrogenic division, as the two daughter euglenae are mirror images of each other. So the symmetrogenic division is very, very important. The term is very important because the symmetrogenic division is nothing but as the division of these your two daughter nuclei are mirror images, which means exactly the same. That is called as a symmetrogenic division. What the given account of the pseudopodia. Okay, so the pseudopodia and temporary extensions of the cytoplasm, which is found in the rhizopods. Okay, so this pseudopodia, this is very important. So in case of amoeba, so this is the amoeba. Consider this is an amoeba. So there is an external projection. So like this. Okay, so these are all the projections. Finally, the amoeba will be look like this. Okay, so these are all the external extensions of this amoeba. So this extensions is called as the pseudopodia. It is found in the rhizopoda. Okay, rhizopods. So they developed in the direction of the movement and helps the organisms move on the substratum, functioning like legs. So that you should know that the extra extension from the membrane is called as a pseudopodia, and this is responsible for movement of this particular organisms. So we are having different types of pseudopodia: lobopodia, filopodia, and reticulopodia, axopodia. So this is very important. So the lobopodia is nothing but based on the structure of the pseudopodia, we are named the podium as this lobophilo, like that. Okay, so if the pseudopodia is blunt, finger like, so if they are in the structure, we are calling them as a lobopodia. We can call this lobopodia in case of amoeba and entamoeba. Okay, so filopodia in the sense it is very thin and fiber like structure. It is seen in the euglypha. Okay, so that is why we are calling them as the filopodia. Reticulopodia in the sense this is all the net like. Okay, so net like structure, we can see them in the reticulopodia, Elphidium. Okay, axopodia and helopodia. They are radiating like sun rays seen in the actinophores. Okay, so the axopodia, it is also called as a heliopodia. It is radiating like a sun ray. It's a sharp like structure, so that can be seen in the actinophores. So that is why the name axopodia and heliopodia. See, in this here it will be very blunt like structure, so that is why it seems like a lobes. That is why we are calling them as a lobopodia. Philopodia, it looks like a fiber. Okay, so that is why philopodia in case of euglena. Reticulopodia, it is like a net like structure, so that is why we are calling them as a reticulopodia. Axopodia, it looks like a sun rays. Okay, so that is why axopodia, it is seen in the actinophores. Formation mechanism, it is formed by the conversion of gel to sol and sol to gel. Okay, so these are all the formation mechanism, how the pseudopodia is getting developed. It is the theory, the name of the theory is called as the sol gel theory, sol gel transformation theory. Okay, gel to sol and sol to gel. Okay, so this is what, how when the gel is converting into sol and the sol is converting into a cell. Okay, gel is nothing but it is a viscous outer endoplasmic part, and the sol is called as, it is fluid like inner endoplasmic part. Okay, and during this conversion there will be an extension. We are calling them as a pseudopodia. So examples of the pseudopodial movement is amoeba, polystomella and actinophores. Okay, so these are all the best examples. So another example, another thing, a question will be this: draw a neat labeled diagram of euglena. So you have to draw a neat labeled diagram. It is not necessary to draw the exact picture, and it is impossible. So you can draw a basic diagram. Okay, so this is an outer ear, so we can call this as a cytostome. That is nothing but a mouth. Technical word of a mouth is cytostome, and there is a long flagellum, and it is a stigma, and this is a reservoir, and here it will be a contractile vacuole, and this is what a blepharoblast, and here we can see the chromatophore, paramylum, myonemes. This is the nucleus, and the outer covering is pellicle, and inside the substance is called as the gel-like structure is called as a cytoplasm. There is a short flagellum is also there, paraflagellar body, and these are all the important structure we couldn't neglect in case of picture. Okay, so next the question will be a diagram of this paramecium. Okay, and definitely you have to mark the point macronucleus and the micronucleus. It is very important, and the oral groove. See, so this structure is the oral groove. Okay, so and the mouth-like structure is called as a cytostome, and this structure is called as a food vacuole, and this is cytophage. The outer layer is called as the cytostome is related to mouth, cytophage is related to the anus. Okay, and there is a contractile vacuole. All these were the important things you shouldn't neglect. Okay, so that's all about the short answer question in this chapter. We'll meet you in the next chapter. Okay, thank you so much.